Welcome to a new episode of People and Science Live with Karger. We are here today at the home of a very special guest, a Nobel Prize laureate, Dr. Jules Hoffmann. Thank you, Dr. Hoffmann, for having us in your home, in your study.
My pleasure.
We're having a special recording today in person with Dr. Hoffmann, whose groundbreaking work has left an indelible mark on immunology, and is the recipient of the 2011 Nobel Prize in Physiology and Medicine. And we're going to talk to him a little bit about his journey in research, about immunology, his impact into the world, and how it feels to be a Nobel Prize recipient. So thank you again and welcome to this episode.
Thank you very much. Looking forward to this interview.
Oh, me too, absolutely, and I'm sure everybody as well, researchers, librarians, and all our followers. Let's start from the beginning, right? You made this groundbreaking discovery in immunology. Can you tell us a bit about what initially drew you to the field?
Yes, let me first tell you that every Nobel laureate says that, which is true. The prize was given for the work of a whole group. We were many co-workers, and over the years, I think about 50 to 60 people have worked in the laboratory, and I want to acknowledge, first of all, their contribution. So, why did I start working on insects? Well, I was born in Luxembourg, and my father was a professor of life sciences in college. And he was very much interested in what we would call today biodiversity. Mm-hmm. So we went to the field, recording various types of insects, of various bugs, for bloodsuckers and so on. And he took me with him from the age of 15 on. And I found it was very interesting, and I was very excited about that. And he asked me to write down my results, particularly about grasshoppers. Okay, so I did that, and we even succeeded to publish it in the annals of or the archives of the in Luxembourg, the academy, the small academy of Luxembourg, and so I published with him about 5 papers or 6 papers at that time, in these archives of the Luxembourg Academy of Sciences, and then in Luxembourg at that time, so. I left Luxembourg in 1960 because there was no university at that time. Now they have a university. Mm-hmm. And so I selected Strasbourg. We had the freedom of a choice, but I selected Strasbourg because it was close to Luxembourg. And also because I love the atmosphere. We visited once Strasbourg, I love the atmosphere here. But I got a proposal by one of the professors in zoology. I was in the Department of Zoology at that time, and he proposed that I do a PhD work in his laboratory, mm-hmm. and he was working on grasshoppers.
Okay.
So I felt quite sort of at home.
Yeah.
And also, as I mentioned, what I had done was biodiversity. It was not physiology. And so, I accepted this position, which was a temporary position, and that must have been in 64 actually. And he had worked on the control of the development and reproduction of those grasshoppers. Okay. And particularly, they did only at that time, there was no molecular biology at that time, speaking about 64. Mm-hmm. And so he said that what they had done was histology, cutting sections through the animals, yeah. and then transplanting supposed endocrine glands from one insect to the next, and looking at the effect on development or reproduction. Mm-hmm. Okay, and that was, so he told me that they had at that time, they thought they had finished their job. They had identified all the putative glands in the grasshoppers. They had transplanted everything, looked at all the effects, and they said, that's not, for you, as you are starting your thesis, that's not a real future. But, I said, when I did all my experiments over those last 20 years in Strasbourg for him, I never took any precaution of asepsis. So I never put any, well, even the tools which I used to transplant tissues and organs, I didn't sterilize them.
Yeah.
And then I put back the animals which had undergone surgery, I put them back into a non-sterilized cage or environment, yes.
Yeah.
And he said, I never, when someone asked me, so what happened? Did they develop diseases? I never noticed any infection going on. So he said, I think looking at the reason, mechanisms of the fight against infections in grasshoppers would be something very interesting, and nothing is known about that. But he hadn't studied what had been published already.
Okay.
I'll come to that in a moment. So I say it's really a totally new field. Yeah, I was at the time I was very excited about the idea, and then I realized rapidly that there had been quite some developments again. And then he was not an immunologist, and
Right.
He didn't know I was not an immunologist either, because at that time immunology was not taught in zoology courses.
Yeah, of course.
You had to go to medical school, and so it was really it was something very dangerous into which we got involved, and then and he didn't really follow my work. At that time, there had been two very important scientists in immunology. Immunology really started only at the end of the 19th century. Before that, number one, when people got infections were going on all the time, but no one knew exactly, number one, why there were infections.
Yep.
Most of the people, it was classically people were told classically that was punishment for sin, by God, and so on. And so it was only with Pasteur in France and Robert Koch in Germany that it was really demonstrated that the origin of infections were bacteria. So it was not before that. You imagine when we started in the 60s, as I said, there was we didn't really know. I studied all those systems, and then I started working on the grasshoppers. I saw that it was phagocytosis, and it was fine, and but that was not really new. It was still, so we put some details, we added some details, new details, but that was basically it. And then there had been one report in the 30s about the presence in the blood of challenged insects of soluble factors.
Okay.
And so, but that was not continued. So what we did, I'm passing now, I did my PhD work then here in Strasbourg on the blood cells and how the various aspects of in blood cells. That was it. But then came the period of that when it really changed the stories was in the 80s. So that took a long time. During that time we worked on other aspects of biochemistry, of steroid hormones and so on. So we were occupied, we got funded and so on. But when my Pierre Joly, who gave me the topic, when he retired, and so that was in 78, he was a professor at the university, and so they had to find someone to step in for that, and he had proposed that I would get that job, and then there was a strong opposition from the side of a certain number of professors in the university saying, now look what he does, working on insect defenses, that's for agronomy, but that's not for a fundamental university study and so on. And so they wanted us to move to some other place. And finally the president of the university, whom I had come to know as I gave several lectures at the time, and so he proposed a compromise, and he said, okay, let us, the lab was relatively big, much space, many technicians, and so many people, we were 15 people at the time. So they said, well, let us ask Hoffmann to continue being a scientific director, not a professor.
Okay.
And then on the other hand, we leave him 3 years, and in 3 years we'll see what we do.
What happens?
Yeah. Those 3 years were fortunate. We then really found that there was we left the grasshoppers for flies for various reasons, which I'm not important enough to go into this, but we couldn't on grasshoppers, we couldn't do no genetics. And at that time, what had changed the whole view is that there was molecular biology there, and then molecular genetics also, and all that was impossible to do at that time on the grasshoppers. And flies, it had been flies since the beginning of the 20th century. Had been the work of Tom Morgan, had been 2 Nobel Prizes, one to Morgan, one was student Mahler. And so we were able, we were collaborating with a very good group in Strasbourg doing analytical chemistry. And then over a relatively short period we identified the peptides. We had no innate immunity. We had no reason to believe that innate immunity, adaptive immunity was coming up. I'll come back to that in a second. So we have similar molecules which are induced. That's one point, but that was it. And yes, I should mention at this moment here that already in 1960s or so, at the same time when we were still thinking only of innate immunity, Jerry Edelman and Rodney Portner had established the structure of antibodies. Okay, and that was a big, they shared the Nobel Prize also, and they were happy about it. And so that asked the question, where are those antibodies produced? That was not known at the time. And then a whole series of very distinguished scientists worked on, continued, tried to find what was the origin. And then it was found in the 70s, people like James Govans and Max Cooper, and Max Cooper, they found that there were 2 types of lymphocytes, the B lymphocytes and the T lymphocytes. Yes, T for thymus and B for bursa fabricius or bone marrow.
Bone marrow, yes.
They were discovered actually in the bursa fabricius in birds by Cooper first, and then it was extended to a mammalian system bone marrow, so it's kept C and B. Okay, those things were happening when we were in this stage. And no one really thought that it was serious to continue working on flies. It's not immunity, flies. It's just antimicrobial peptides. Forget about it. And then what changed, Stephen, let me mention, this was in 1970, which is relatively recent. I was head of the laboratories, that was relatively significantly involved in the administration, let me say, in the juries of the CRS, so I had some influence there, and I had very good contacts to directors there, and then we had to renew our institution, and that seemed to be without any problem. And it was without any problem, but then when I told the director, I will never mention his name because he's dead now.
Okay.
And so I told him that we had decided we would stop all research except for immunity. And then he said, don't do that. Come on, it's not immunity what you're working on. And I say, look, we have those antimicrobial peptides now. They're also present in humans, but antimicrobial peptides, that's something totally different. You have no flies, insects have no real immunity. Okay, and then, I was fighting to end in the end, he was tired of my comments, and he says, okay, do you really want it? And I say, yes. Okay, Jules, go ahead.
I just wanted to go back to your research journey, and you mentioned that you started publishing with your father when you were quite young. were you around, what age were you?
17.
17. I mean, that is quite impressive, and I'm sure a lot of researchers are watching this. And do you ever felt this push of publish or perish during your research career?
Yeah, I mean,
the need
everyone,
to publish a lot.
yes, everyone feels that, and you know, well, maybe we'll speak about that a little bit later. I have a laboratory now in China, and there's even, it's worse even than,
Really?
yes, in the US it's terrible, and it was not like that when I started here in France.
Oh, really? Okay.
They were softer,
Mhm.
but now it's exactly the same. It's and that's it's both good for science, but it's a pity for the researchers, because you have a tendency, if your career depends on that, if your salary depends on that, and so on, then you select topics where you are sure that something will happen.
Yeah.
For instance, if you are a medical person and you say, well, I have this type of disease in this patient, let me show if there are also toll receptors involved, you can produce a paper relatively quickly.
Mhm.
And if it's published, it adds to the list of your papers.
Right.
I mean, it's unfair if you block beginnings which are not really producing good papers.
Right.
If you judge a topic or a person only on the results, only on the publishing results and the journals which they invest, I think it's not good.
Do you think it would have been, like, if you were to do your research journey with the research you've done nowadays, would you have had the same sort of pressure? Do you think it would have been worse? Would you have been able to jump from something that was seen as agronomy or zoology to immunology?
It was possibly in my time.
Yeah, but what about now? Do you think it would be still possible to have
I
this
don't
blue
think
sky
so. No.
research type of thing?
No, I'm afraid not.
It's a pity.
I know. I feel like now as a lab you can do it. I mean, if you have a good lab, a large lab, then you can say, okay, you do this and the other one does that, and then we put the names of the two people. You can arrange your atmosphere like that. But no, it has become very difficult.
And connected to that, we talk about, Karger obviously is a publisher in the health sciences, and we talk a lot about open access and science communication. That's something that is really, really important nowadays in the scholarly community. The reason behind open access being this important is that it would allows everybody around the world to read any article without having to pay for subscription, right? But you pay to publish. This would have maybe helped, let's talk about the 60s, for you to see what was going on in a lab around the world, like very much, easily, just to see, actually they're doing something that is relevant to what we're doing. This is like some preliminary discovery things. So what is your take on open access? Let's hear.
I mean, my response is double-fold.
Yes.
Number one, at that time, yes, it would have certainly helped, that's for sure. But there was still, we had many meetings at that time.
Mm-hmm.
Traveling was not as complicated as now, not as expensive. You see, now, you have, I have every week I have Science and Nature coming home to my place here, sell every 2 weeks. And so, that has become so much easier than reading the papers. Papers were difficult in that time, in the old times. But we had many meetings, and they were essentially in Europe. And I started going to the US the first time, I think it was in, I don't remember, 75 or so. But okay, so I would say the communication is very good nowadays. And of course, open access, communication doesn't give you access to, not always, to the whole paper, to material and methods, and you want to read also that and see the graphs. For that it's important to have open communication.
Yeah, absolutely. I think we're, as a publisher, but all the publishers as well, as part of the open science movement, we try to ask authors, researchers to share their protocols, their methods, their data, make everything because we want to make sure that it's can be reproduced
Yeah.
anywhere else in the world and build on top of those
Yeah.
results. So I think that's one of the most I think important things.
I
to
agree with you. That's very good.
And as part of that, I saw in an interview that you did a while ago that you were saying how you were talking about the importance of basic research and also your responsibility as a Nobel winner, but also as a researcher to make sure that you reassure people that science is a good thing, is trustworthy. Now with open access and science communication, we are trying to get across some messaging to the people, to the public that cannot read scientific articles. As we said, they're very complicated to read. There's a lot of very technical terms. What do you think scientists should do to regain the trust of the public that is been going a bit to lower levels since the pandemic, for example?
Yeah, well, that depends. For the last, as regards the last point, I would say that depends on the country and on the population. I mean, the polls in France have continuously shown that there's a big trust in science here, about 80% or so.
Oh, wow,
say.
okay.
Yeah, but I agree that what happens in the United States is scary. But what can you do? You can tell people, they are stubborn. You can tell people, look, for instance, I'm reading New York Times every day, so they say that now in the United States, we're speaking about Kennedy and this attitude, in the United States, there's a difference in survival to COVID when the governor is a Democrat versus a Republican.
Oh.
It's unbelievable, you see, because in the importance of politics on this attitude. I mean, when you say, for instance, also here in France, just 2 weeks ago we learned we have again an important flu disease, important level of people who suffer from flu disease, and some of them die, and they told us that in the hospitals 70% to 80% of people who get hospitalized with flu have not been vaccinated against flu. We have a good vaccine, everyone has a good vaccine against flu now, and so it's so obvious, but they don't want to.
But what do you think, like more broadly, without going to I know the difference between countries is there and it's very much connected to politics, right?
Yeah.
But more broadly, how do you think the scientific community can respond to this kind of misinformation that is also made bigger and wider by social media and media outlets that kind of can take any paper off the internet that is not even being peer reviewed maybe and just share it? Well, yeah.
I mean, it's a question which everyone asks, and the only answer I see is it's a matter of education. And the problem there is that, there's one very big difference between people who doubt everything in that case and people who are scientists in that case. So what is there's one main difference. The people who are not convinced, and so are people who are used to accept miracles, to accept revelation, to accept the idea that Moses was one day saying
anything that can help them.
Whereas scientists only accept what they can experimentally prove. And in science, you see, for instance, I am not even scared by there are some people who cheat in science, but it's minimal. Because in science, if you find a result, like the ones we spoke about, well, they immediately either it's interesting and then no one cares, or it seems interesting, and then immediately people jump on it and try to reproduce it, and to extend it to their own interests and so on. And so it's immediately checked.
Yeah, it's
by
the community. Like you have to,
It's so. Who's going to check the revelation of Moses?
That's a good point. Going back to the COVID pandemic, right?
Yes.
That is such a big topic. It's one that has brought basically immunology in the forefront and center of the
Yeah.
public discussion, right? And
Yeah.
do you think that in general immunology is understood by people then? Do you think there's a more attention to it also for investment by, research institutes and governments and things like that? Do you see a change since the past?
Oh, there are many more laboratories working on a very high level on this now, and it's very complicated. I can tell you. I really think, and I know that many more laboratories are now studying this aspect. Not particularly for SARS-CoV-2, but for the idea that other pandemics will arise one day. And so the more we know about the way this pandemic was,
better we can deal with that.
yes, the more we can fight that aspect. So there is, for that, yes, certainly more work going on.
They're taking more notice of this subject area, like or it's just a fad and it will they'll change their.
In my position, I can only speak about the people whom I see, with whom I
course,
talk,
yes.
and that's a community which is convinced already. Like you say in French, prêcher à des convaincus. It's to preach to people who are already on your side.
Yeah, okay.
Yes. And so, but I think France is, well, it was, for many years it was still very open. But now the social networks are really creating very bad atmosphere. I'm afraid,
Yeah.
yes. Everywhere, in every country, but particularly, of course, in the United States.
Going just back to research and the exciting side of research, I think, like, from your experience, you must have had this excitement to find something and to keep digging and across the kind of research you were doing. Nowadays, there's a lot of use of AI. We're talking a lot about AI, new technologies that can help researchers in their endeavors. They can help also publishing their research. I'm thinking, do you think that if AI gets to a point, and is already, I think, at that point, where they can scan the body of research in a certain discipline, let's say immunology or innate immunology, and find already the gap that needs studying, say it starts collecting all the pieces and say, oh well, this is an area that hasn't been studied yet or explored yet. So the researchers know already where to go directly. Do you think that takes away a bit of the magic of research as you know it?
Well, we have not done that, and I'm using AI in a very simple way. So in which year was done this, and when was done, how many people have, and so how many people have. Oh, for instance, that's an interesting point. For instance, I have asked IA the other day, AI, AI, how many people, how many papers were published in the various years, having in the title Innate Immunity, and in the title or in the abstract, but only that, not in the citations. So, and then you see, it's unbelievable. I'll put that, I'm just writing a sort of biography, and it's unbelievable that you see there were years after the Second World War, there was no paper published, and then 1, 2, 3, 4, and so on. And then after the push, which we mentioned just, we went up now 3,000 to 4,000 per year, and there were some of those which have toll receptors, and their title or abstract, it's more than 70,000.
Wow, that's a lot of people.
And it's only to show the interest. I'm not saying the importance, but this is what people are.
But that's a good segue for one question that I have, because you serve on one of the Karger's journals, which is Journal of Innate Immunology, and before its launch, there were a few journals on immunology, but nothing specific on that, and then it kind of, as you said, more and more articles were published in that subject area, so it was a bit overlooked up until the 60s as a subject and a discipline. Do you think there's any other discipline or subject nowadays that's been overlooked and you've seen, I've seen that happening when I was doing research and now it's happening for a different subject area, or something you noticed? that is it.
Yeah, I think I was expecting such type of question from you, but it's relatively difficult to say. What's something which I would, if I were to start again,
Mm-hmm.
I would probably, I'm working, as you know, in China now.
Yeah,
We
yeah,
have very
of course.
interesting data there, but that's still in the fly, and that is looking at the use of early developmental genes,
Mm-hmm.
and then in immunity against cancer this time.
Oh wow, okay.
And that's
It's very
okay. That excites us, and so more generally speaking, I think what's probably going to be very important in this sense. You're right to mention this intelligence, artificial intelligence, is that, for instance, I've read this morning a paper here. They say it's so complicated for cancer because you have, of course, the cancer we now know. I'm speaking here about humans.
Mm-hmm.
This medical, and I'm not a medical person, but I'm following that field. They say we have the cancer cell. Cancer's a whole group of cells. There are many different cells. There are cells expressing TLRs,
Mm-hmm.
and there's the lymphocytes of various subtypes and so on. And it's very complicated. And then to see, and some of these can be blocked in this new system, checkpoint inhibitor.
Mm-hmm.
You see, or in CAR T cells, and others cannot. And maybe this intelligence will help. That you can do now sequencing cell by cell.
Right.
It's single, so even we do it in the fly,
Mm-hmm.
so single cell sequencing, and then you can, if you improve the technique and that is being done, you can say, yeah, well, there's so many cells which are the subtypes which normally react to, and others who no, that doesn't help.
Mm-hmm.
So I'm convinced that at this level the artificial intelligence is going to make a big difference.
Yeah, as a tool to help you
Yes,
out.
as a tool to. You have to ask. I mean, our own role as scientists will be to continue asking good questions, and then the rest we are dependent on
Mhm.
myself. If I look over my whole career, the question was good.
Yeah.
It was not even asked by me, but by my supervisor. The question was good, but he didn't follow then.
Mm-hmm.
But we were dependent on the progress of the techniques. Without the molecular biology, we would not have been able to.
Yeah, it's
do
all
what we did.
interconnected, of course, and the curiosity is a
Yes.
very important part
Yes.
of that, and that's why I was just curious to see if AI takes away the curiosity by already mapping a way of
No,
things
it
that you need to
simplifies
explore.
your
It
work,
just
I would
simplifies
say.
it. Okay.
Well, it's maybe too much to say. It just simplifies it, but it gives you a tool to access things which are impossible before
Yeah.
that. Yes.
It makes process quicker
Yes.
as well. Yeah.
Yeah, absolutely. More expensive, but quicker.
Yes. Looking to the future, I'm not even going to ask you in 10, 20 years, but even just in the next 3 years, let's say, do you see any exciting development happening that will have an impact?
Yes. I think, I'm sorry. Oh, no, it's
Hesitate.
what if I were to select something, it would be continuing immunity, and now innate immunity. For instance, we suspect
Mm-hmm.
that all our defenses are controlled by innate immunity.
Mm.
I mean, the initial point comes from the recognition. The first signal is a signal of innate immunity
Right.
at every level in the tissues. Now we look at the at the immune system as one big part of the body, and so there's no place in our body where an aggressor, or for that matter, a misformation is not sensed by a cell of the innate immune system. And that will then give the signals, so there's many signals, and that will stimulate the adaptive immunity, which has, of course, the hallmarks of specificity and memory. Coming back to immunity, in general, what is now really the big questions are, we know now that the immune system is involved also in neurodegeneration.
Mm-hmm. Yes.
And now, what are the molecules which we know the TLRs are expressed there?
Right.
And but how does this go? We don't know. It's then people speak about the amyloid plaques or the tau tangles and so on. That maybe so, but how to prove it?
That is connected.
Absolutely,
And
absolutely. And the microbiome has a strict effect on the brain.
On the brain, so it's all interconnected.
It's not that there's a direct connection like that, but it's metabolites produced in the gut, which go into the blood and the gut, come up to the brain, and they can have a positive or negative effect.
Yeah, and the triggers, from stress or anything that comes in, has an immune system response. We can close with a nice, a bit like lighter question, if we will. So, Isaac Asimov, stated that the most exciting phrase to hear in science is the one that heralds new discovery. It's not eureka. So I found it, but that's funny. Would you agree with this statement? Would you also say? Did you say anything like that when you discovered that your research had that moment of genius, of eureka?
Well, no, I'm going to disappoint you. I was often asked that question, and also related to that, what was your reaction when you got the Nobel Prize? And I say it was really an enormous relief, because all the ideas which you have been developing and thought they would lead to something, finally they did, and it was recognized. And so there was the recognition by the Nobel Committee that this was an important result. That was for the lab, for our lab, that was really rewarding. And so in essence, what we have demonstrated, and we as a lab, you see, is that insects not only have a very active innate immune system, but they can be used as a model to study immunity. And that was really not accepted until 1990 or so, when I got, and that's probably one of my best merits. I'm not the only one. In Stockholm, they also, Bowman did something like that, but then he stopped and he went over to other things. But my lab is going, my former lab is going to continue now working on antiviral defenses.
Yes. Okay. Thank you so much for answering all these questions and for all your lovely answer. And it's been a pleasure having you on this episode.
It was a pleasure for me.
Thank you.